All right, let me show you one example. It's the first gene for which alternative splicing was discovered, and it's been studied in some detail because of that. Um, it's the calcitonin gene, which is called calc-A, but strictly speaking, this gene produces two important proteins. One is calcitonin, which is an important hormone that regulates calcium in the blood. It's produced in the thyroid. Another is CGRP. It's produced from the same gene. This peptide is calcitonin gene related peptide, and it's expressed in neurons, and it has some functions in vasodilation and pain reception. So this is one gene though, right? So this isn't just the calcitonin gene or the CGRP gene, it's both. It's called calc-A. So the peptide structure then would look like this. So backing up a bit, we've got the structures of the genes here from the UCSC genome browser. These are different ways of annotating the same piece of the chromosome. For each type of transcript that's produced, the start codon is in the same place because the transcripts have the same three first exons. The start codon's in exon 2, so here's coding region, but then the terminal exons are different. So that leads us to a situation where the calcitonin peptide looks like this. It's got a blue part and a red part that I've color-coded here, but the CGRP peptide is different, meaning the blue amino acids are the same, but the green amino acids are distinct from calcitonin. And so they have different functions, but they're also produced in different tissues too. So how is this regulated? So what I'm telling you is that the thyroid exclusively makes this splice form, and neurons exclusively make this splice form. How is this regulated? This has been studied since and is a pretty neat little story. So let's imagine these are the primary transcripts for this gene, but this one is going to get spliced with this exon, which is the last exon, so it's polyadenylated, and this will lead to production of a calcitonin peptide. Here is the same thing for CGRP. So what I'm telling you is that in CGRP, this whole intron gets spliced out, and these splice junctions, the splice junction here gets ignored, as does the polyadenylation signal, because this is the last exon of this transcript. And then the splicing machinery just chooses this exon to splice into, and the RNA polymerase doesn't terminate until this exon down here. So what's going on in the thyroid, apparently, is this. First of all, we have normal recognition by the spliceosome of this splice acceptor site. There are also some proteins that bind inside the exon of this RNA molecule that promote the recognition of this splice junction. It stabilizes it. So these are in addition to things that we find in the spliceosome, which is partially denoted by these orange and red circles here. There is another protein, TIAR, which binds to a U-rich sequence in the primary transcript, and this apparently promotes polyadenylation at the site just downstream too of this exon. So when you put both of these together in the thyroid, we wind up splicing into this exon and then polyadenylating here, and that's it. The transcript is completed. Now, in neurons, a few different things are happening. First of all, there are proteins in the FOX family of RNA binding proteins that bind at the end of what would be this intron, where the splice acceptor would be in the thyroid, and they specifically block splicing from happening at this location. Furthermore, there's a protein that is in the HU or HU family of RNA binding proteins that also binds to this U-rich sequence, and it displaces the binding of TIAR, which is trying to encourage polyadenylation, but this does not happen. So the combined effort of these proteins means that splicing doesn't happen here, polyadenylation and termination doesn't happen either. And so the primary transcript continues, and the splicing machinery is free to splice into this acceptor site. It also winds up splicing these two exons together, and then finally termination and polyadenylation happen down here. these proteins are presumably expressed in a neuronal manner, or at least regulated in that way, which explains the tissue specificity of this process.